Alright, so this is the first of 5 lectures for fatty acid metabolism, and in this first lecture we're going to give an overview of fatty acids, the different steps involved in fatty acid synthesis and fatty acid breakdown, and then we're also going to cover ketone bodies. So first, just an overview of lipids. Fatty acids are the simplest lipids, and they're composed of unbranched hydrocarbon chains with a terminal carboxyl group. So if you come down to the figure here, here's that terminal carboxyl group, and then as you can see, these are the unbranched hydrocarbon chain. So just single bonds here between each of these carbons. That would be saturated. You can also be unsaturated, where you have a double bond thrown in here as well. So saturated is when you have single bonds throughout all the carbons. Unsaturated is when you have a double bond. It could be one or it could be many more. And remember, from organic chemistry, you can have cis double bonds, where you have both the R groups are on the same side. And then you can have trans, where the R groups are on opposite sides. And this corresponds to that what you hear about in popular culture with trans fats and cis fats. You're trying to avoid trans fats. So fatty acids are oxidized during the fasting state. So fatty acids are typically stored in adipose tissue, and then they're kind of stored for a rainy day. When you go through prolonged fasting, you break them down and they generate a significant portion of the body's necessary energy. Fats actually pack a lot of energy. Most fatty acids are 16 to 20 carbons long, and they're sorted into 3 classes. So you have the short chain fatty acids that are 2 to 4 carbons, and then you have the medium chain fatty acids that are 6 to 12 carbons. And these are directly absorbed in the small intestine and then can also diffuse freely from the cytosol to the mitochondria without the carnitine shuttle. And the significance of this statement is that these medium chain fatty acids are able to cross lipid membranes, both at the small intestine level and then also in the mitochondria as well, and they don't need this shuttle. Versus long chain fatty acids, which are 14 or more carbons, they are found in triacylglycerols or triglycerides, which are 3 fatty acids bound to a glycerol, and they do require the carnitine shuttle because they're not able to pass through the lipid membrane unassisted to transport them from the cytosol into the mitochondria. And the significance of that is that you would want to transport them to the mitochondria to undergo beta-oxidation, which is where you would break them down to then generate the energy you need from the fatty acids. This is a nice diagram here where you show this is glycerol, this 3 carbon structure here, and then it's bound to 3 fatty acids. 1, 2, 3. This is a saturated, because remember each of these represents a carbon attached to a hydrogen, that it's all single bonds, versus these are unsaturated. And these would be cis, unsaturated, because these hydrogens would be out here, and these are on the same side. Same thing here. These are all cis bonds here. These are all cis to each other. So the overall process of fatty acid synthesis occurs mainly in the liver and in adipose tissue. Makes sense. The liver is, a major hub of metabolism, and then obviously adipose tissue is where the fatty acids are eventually stored. Dietary carbohydrates in excess of that needed for energy production and glycogen synthesis are converted to fatty acids in the liver during the fed state. So what that means is, you consume a large carbohydrate meal or a meal full of high amount of carbohydrates. You're going to break some of that down for energy. You're going to take the first excess of that and form glycogen. But once you've maxed out your glycogen, then you take what's left and convert it into fatty acids. And the way that occurs is you break it down into acetyl-CoA, which then, as you follow the arrow up here, can be converted into fatty acids. Combine that with glycerol, and there you get triglycerides. You can also have acetyl-CoA be contributed from amino acids from protein breakdown as well. That's another way you can contribute towards fatty acid synthesis as well. And the other thing is fatty acid synthesis, as we say here, occurs in the cytosol, and it uses acetyl-CoA. So now we'll go through the 2 phases of fatty acid synthesis, or the steps. So the first phase here is you've got pyruvate here that you've synthesized as a result of glycolysis. Remember, that gets transported to the mitochondria from the cytosol, where it gets converted to acetyl-CoA, our starting point for fatty acid synthesis. And remember, it can combine with oxaloacetate to form citrate, which is catalyzed by citrate synthase, which is the first step in the citric acid cycle. So citrate can continue on through the citric acid cycle, but it also can go up here, where it can then be exported out of the mitochondria into the cytosol via the citrate shuttle, which would be represented here. So citrate gets exported out into the cytosol, and then it's converted back into acetyl-CoA and oxyl acetate by the enzyme citrate lyase. And so the purpose of that was to get acetyl-CoA back out of the mitochondria and into the cytosol where fatty acid synthesis occurs. And the reason this would get shunted is because in the fed state, if you've just consumed a very large carbohydrate meal, at a certain point the flux through the citric acid cycle is going to stop. You're eventually going to get a buildup of citrate, and that's going to shuttle it from going through the citric acid cycle to going through this process to where you convert these excess carbohydrates into fatty acids. So now that you've converted it back to acetyl-CoA and oxyl acetate, acetyl-CoA can then undergo conversion to malonyl-CoA, which is the first committed step of fatty acid synthesis, and this is catalyzed by the enzyme acetyl-CoA carboxylase. This is an irreversible reaction, hence committed, and it requires the biotin as a cofactor. What's significant about malonyl-CoA is that it can inhibit carnitine acyltransferase 1, which we'll talk about later in the lecture. This blocks the transport of newly synthesized fatty acids across the inner mitochondrial membrane into the matrix where fatty acid oxidation takes place. So what's the significance? So with fatty acid synthesis, you go through a series of steps to form palmitate, which is a 16-carbon fatty acid. And we'll go through these steps on the next slide. The point is here is you've produced palmitate, which is a long-chain fatty acid. If you were to want to get that back into the mitochondria, you would need this carnitine acyltransferase enzyme to then bring this back into the mitochondria where it would undergo fatty acid oxidation. Now, you don't want to do fatty acid oxidation because you're already in a high energy state. You've consumed a lot of carbohydrates. They've been used up to produce glycogen. You've produced enough energy. Flux through the citric acid cycle is slow. So now you want to convert that excess glucose into fats where you can store them in adipose tissue for a fasting period, where then you could break them down for energy when carbohydrates are low. So you shunt citrate to this way, back out into the cytosol, you create acetyl-CoA, it gets converted to malonyl-CoA, you've just created fatty acids. Now this would be a futile cycle to then just send these back into the mitochondria to can be converted into eventually ATP, which can be used for energy. So you want to block. So malonyl-CoA comes over and blocks this and prevents this from happening. So then you can take these fatty acids and convert them into triglycerides and store them in adipose. So we'll start over here with acetyl-CoA. Acetyl-CoA gets converted to malonyl-CoA by acetyl-CoA carboxylase. It essentially has a CO2 added to it. So then you have formed malonyl-CoA. And then the rest of fatty acid synthesis is going to be carried out by an enzyme called fatty acid synthase, which is very complex. It's almost like an enzyme complex that carries out multiple different types of reactions. And so it continues by progressively adding 2 carbon units from acetyl-CoA until it reaches palmitate, which is 16 carbons long. So you keep going through this process where you keep adding the chain, and you have this chain attached to a carrier protein here, an acyl carrier. And as you go through the process, you keep breaking down to acetyl-CoA, and then acetyl-CoA gets broken down to malonyl-CoA, and these are, getting fed from, the excess glucose consumption. And so this ends up, providing a pool of acetyl-CoA from that excess, glucose consumption. And that acetyl-CoA keeps getting added to this growing chain and keeps adding 2 carbons to this growing chain until you reach palmitate. And so the end goal here is to produce palmitate, which is 16 carbons, all single bonded. And so the way this process goes is first, you undergo a condensation, so you remove a CO2. Then you undergo a reduction reaction, and your reducing agent is NADPH. Remember we talked about this in the pentose phosphate pathway? Well, here's NADPH in action here. So what you're going to end up doing is reducing this bond right here. And so you can see that here, where you have this hydrogen here and this hydrogen here. And so the next step there is then to undergo a dehydration reaction. This is number 2. So you remove this water, and that gives you this bond here. And then you still have this double bond. So you're going to reduce that again using another NADPH as your reducing agent to give you this final product here. And then here, this process, you come back and repeat itself, and you keep adding on 2 carbons, 2 carbons, 2 carbons, until you eventually get to 16 carbons, which is palmitate. And another important thing here is notice there's 2 equivalents of NADPH used here. So fatty acid synthesis definitely uses up a good portion of the NADPH in the cell. And is definitely, as a result of that, is definitely regulated by or responsive to the NADPH and levels and the NADP levels, or that NADP/NADPH ratio within the cell. And so again, this NADPH comes from 2 sources. One, what we said, was the pentose phosphate pathway. The other source is that if you come back here with the citrate shuttle, when you're converting citrate back to acetyl-CoA oxalo acetate, so acetyl-CoA goes this way towards fatty acid synthesis, but the oxalo acetate then goes back to form malate, which is catalyzed by malate dehydrogenase. And then malate gets converted into pyruvate, which uses up an NAD+ to produce an NADPH. And this is catalyzed by the enzyme NADP malic enzyme. So that's the other source of NADPH for fatty acid synthesis. So during the fed state, when you've just consumed a big meal, body's digesting it, metabolizing it, excess glucose is converted to fatty acids, and then those fatty acids are combined with glycerol 3-phosphate to form triacylglycerols in the liver and in adipose tissue. The source of the fatty acids are chylomicrons, which absorb fatty acids in the small intestines, or VLDL. So the source of the glycerol 3-phosphate is converted from dihydroxyacetone phosphate during glycolysis, or it's formed from glycerol itself by glycerol kinase. And so if you see here again, you have this excess glucose. It goes towards glycogen. When you've maxed that out, you end up forming acetyl-CoA here. And then acetyl-CoA gets converted into fatty acids through fatty acid synthesis, that process we just talked about. And then it gets combined with glycerol to form triglycerides. Now, an important thing to remember is that fatty acids must be converted to fatty acyl-CoAs by fatty acyl-CoA synthetase. 3 fatty acyl-CoAs are then added to glycerol 3-phosphate. So when you do this, you're essentially charging it so it can then interact with glycerol 3-phosphate to form triacylglycerols. When formed in the liver, triacylglycerides are packed into VLDL, which then can transport these fatty acids to peripheral tissues where they can be used for energy. Triacylglycerols synthesized in adipose tissue, so here are then stored for periods of fasting. So that makes sense. You have this excess acetyl-CoA, gets converted into fatty acids, combined with glycerol, and then you store them essentially for a rainy day, or what I mean by rainy day, a fasting period. So fatty acid degradation is also known as beta-oxidation. And this is where fatty acids are progressively degraded to smaller components. And the ultimate goal here is to form acetyl-CoA, that central component, which can then enter the citric acid cycle, generate NADH and FADH2 that can go to the electron transport chain and generate ATP. So what's important about fatty acid degradation is that it occurs in the mitochondria. So remember, fatty acid synthesis occurs in the cytoplasm. Fatty acid degradation occurs in the mitochondria. And what's important to note about that is cells such as red blood cells that don't have a mitochondria, they can't carry out fatty acid degradation. The process of fatty acid degradation relies on the carnitine shuttle. We'll go over that in a few slides, which brings fatty acids from the cytoplasm into the mitochondrial matrix so that they can actually undergo fatty acid oxidation. So this is sort of a regulatory step to ensure that, again, you're not synthesizing fatty acids in the cytoplasm and then they're ultimately also undergoing degradation, thus preventing a futile cycle. So fatty acids undergo a series of 4 reactions: oxidation, hydration, and then oxidation and hydration again to form acetyl-CoA and a fatty acyl-CoA that is essentially 2 carbons shorter. So minus 2 carbons. And then what happens is this fatty acyl-CoA goes through this process again. So if you'll see, we have fatty acid synthesis here on this side, and then beta oxidation here. So you want to focus on this side here. So beta oxidation is sort of the opposite of fatty acid synthesis, where we had a series of reduction and dehydration reactions. On this side, we have a series of oxidation and hydration reactions. So if we start here, we have this is a dehydrogenation reaction or an oxidation reaction. And what's unique about this reaction is it generates an FADH2 for us. Then it undergoes a hydration, so you're adding a water. Then you have another oxidation reaction or a dehydrogenation. And as a result of this reaction, you produce an NADH. And then the last part here is you have cleavage of acetyl-CoA. So you cleave acetyl-CoA off of that fatty acyl-CoA. And so this fatty acyl-CoA, again, remember, is 2 carbons shorter. And so this part can then go back up here and go through the process again. These two, the FADH2 and the NADH, they can both go directly to the electron transport chain and generate ATP. And then acetyl-CoA can then enter into the citric acid cycle, which then generates NADH and FADH2, which then go to the electron transport chain to generate ATP. So not only do you get FADH2 and NADH as a part of just the beta-oxidation process, you also generate an acetyl-CoA, which then enters into this process to generate even more NADH and FADH2. So fatty acid oxidation can generate a lot of ATP, which is good because it's often called upon during the fasting phase and when you've essentially run out of a good supply of glucose. When you've used up your glucose readily available, broken down the available glycogen stores, then you would switch to using fatty acid oxidation. And so this process continues with this fatty acyl-CoA here. Like we said, it goes up back here and goes through these 4 reactions again, and this continues until it's completely broken apart. So we've talked about ketone bodies in previous lectures, and so now we're going to take a couple slides, though, to really dive in and talk about what ketone bodies are, where do they come from, and how they're used. So they're an alternate energy source, and they're primarily synthesized in the liver, and within the liver, they're synthesized in the mitochondria. And so they're associated with a depleted energy state. So you really only synthesize ketone bodies in periods like this where you have very prolonged starvation. A common example on board exams is diabetic ketoacidosis. This is a form of starvation in a sense because it's more like the body thinks it's starving because there's a lack of insulin. This is typically in patients with diabetes type 1, and it results in an inability to use glucose. So they have glucose, but they can't get it into the cell because they're lacking insulin. And so the body has to resort to other sources of energy, such as fatty acid oxidation. Now, at a certain point, that's going to run its course, and then the body needs other, especially the brain and muscle tissues, need other sources of energy, and that's where ketone body production comes in. So then the body thinks it's even more prolonged starvation because it can't use glucose, so it switches over to ketone body production. And then you end up with this immediate emergency, diabetic ketoacidosis. And then a third example here is alcoholism. And this is where oxaloacetate is shunted towards malate as a result of excess NADH. We talked a little bit about this in the gluconeogenesis lecture. We're also going to talk about this also in the alcohol metabolism lecture in Unit 4, in the nutrition unit. Ultimately, though, during a depleted state, there's an excess of acetyl-CoA, and this mainly comes from fatty acid oxidation. And then this acetyl-CoA is used for ketone body production. Now there's 3 ketones, and here they are: acetone, acetoacetate, and beta-hydroxybutyrate. So as we said, ketone bodies are made in the liver, and this is after breaking down fatty acids, typically. And the way you can remember that ketone body synthesis happens in the mitochondria is that beta-oxidation of fatty acid also occurs in the mitochondria. So we show this here where fatty acids, they get converted into acyl-CoA, undergo beta-oxidation, and give you acetyl-CoA. Just I want to point this out, another minor source of acetyl-CoA for ketone body production is amino acid breakdown. And so amino acid breakdown can result in generating acetyl-CoA. And amino acid breakdown is especially going to happen during very prolonged starvation. So we just want to make you aware of that. So the individual steps and enzymes aren't terribly important. What you want to know is that you eventually end up here with acetoacetate, which is the first of the 3. And then acetoacetate gets converted using an NADH into beta-hydroxybutyrate, which is the second here. And then lastly, acetoacetate can spontaneously get converted into acetone, which is your third ketone body. These 2, acetoacetate and beta-hydroxybutyrate, are the main 2. If things are spontaneously shunting towards acetone, that's typically in a severe starvation state, and things are not good as far as just the overall health and metabolism of the body. So when you have these 2 circulating around, what determines if you convert acetoacetate into beta-hydroxybutyrate is really this NADH level, an NAD plus level. Now, during prolonged starvation, if you remember, beta-oxidation is going to be going on, and that's going to produce very high levels of NADH. And then some of that's going to be used for this reaction to convert acetoacetate into beta-hydroxybutyrate. Now, what happens is these ketone bodies are then shipped out into the peripheral circulation where they can be taken up by peripheral tissues. The main two are muscle and brain. Remember, in prolonged starvation, the brain switches from using primary glucose to primarily ketone bodies for its metabolic fuel. So if you have beta-hydroxybutyrate, if it gets taken up into peripheral tissue, what's the advantage of that is that then you get back an NADH in this tissue that really needs it. And so eventually the goal here is to get to, after generating this NADH, is you eventually convert it back to acetyl-CoA. So you start with acetyl-CoA and you end with acetyl-CoA. Acetyl-CoA then enters the citric acid cycle or TCA cycle, and that generates NADH and FADH2 for you. And both of these then go on to the electron transport chain to give you ATP. And so that's how you come full circle here and how ketone bodies help provide a source of energy for cells. Now, again, if the way the NADH-NAD ratio is going, it more makes sense to just stop at acetoacetate. This can also go out into peripheral tissues. And when it gets taken up into those peripheral tissues, you don't get this extra NADH, but you still get acetyl-CoA. And the last thing here I'll mention is that acetone, like we said, such as DKA, that's a hallmark of clinically of DKA, is you have a fruity breath, and that is usually due to acetone. And so, like I said, if things have gotten really bad, such as in DKA, and you're spontaneously generating acetone from acetoacetate, that's how you get that fruity odor breath. Now, just to come back to this diagram here to just briefly summarize things. So again, the main player here is acetyl-CoA. So remember, you can get there from glucose through glycolysis. The other thing, like we've been talking about, is you break triglycerides down, you get fatty acids, you get glycerol, and this would be during fasting. And then glycerol obviously is going to go to gluconeogenesis. We've talked about that previously to help generate glucose in the liver for peripheral tissues. And then fatty acid is going to get broken down to acetyl-CoA. Now, initially, in the initial phases of fasting, that's going to go to the citric acid cycle, which is going to generate, obviously, again, NADH and FADH2, which will then go to the electron transport chain and give you ATP. Now, if you have a very prolonged period of starvation, then acetyl-CoA can get shunted towards producing ketone bodies. Also, during severe prolonged starvation, you can start breaking down protein into amino acids, and amino acids can then get broken down to, as we show here, acetyl-CoA. And acetyl-CoA, again, can then be used for ketone body synthesis. And again, just remember, the main tissues using ketone bodies are the brain and skeletal muscle. All right, so that closes out this overview of fatty acid metabolism. In the next lecture, we're going to talk about cholesterol and lipid transport.